Hi everyone, welcome back. So, all cells, whether we're talking about prokaryotes or eukaryotes, have to perform a lot of the same functions in order to remain healthy and functional and actually reproduce. All living things have to be able to do homeostasis, and that is a complex, multifaceted process. A lot of things have to be done in order to keep maintaining those internal conditions inside of a cell that are conducive to its survival. This also means that the organism has to be constantly building new molecules, taking resources in from the environment and then using those to build the various proteins and enzymes, lipids, carbohydrates, nucleic acids, those building blocks that make the organism function. There are other things that it may also have to make, including different kinds of toxins and specialized defense compounds, things that help with the organism surviving in its specific environment and facing its unique challenges. It also has to be able to take the resources that it acquires from the environment and break them down into those raw resources, which it can then use to build those materials. In a sense, this is the process of recycling the things that the organism is acquiring, other organic molecules that are acquired by this organism and then repurposed to become something new. The organism also has to acquire energy to fuel these various processes. And so the organism is going to be taking some of the molecules that it acquires from its diet, or in the case of an autotroph, actually build a lot of these molecules from scratch and use those to create ATP. Use those in a process that we call respiration to build ATP. Then we also have other aspects like movement and organization of a cell's interior. And then if we're talking about reproduction, well, that's a very energy and resource taxing process too. Cells have to do a lot in order to survive and reproduce. All of these jobs that the organism is doing require metabolic activity in some form. So we're talking about metabolism here, the chemical processes that an organism is going to be performing to maintain life and do homeostasis. All of these processes, these are all specialized chemical processes that are regulated by enzyme activity. And so there's two main kinds of metabolism that we talk about. We have catabolism, what we might call destructive metabolism, where we are taking resources and breaking them down into smaller molecules, smaller individual components. Then we have anabolism. Anabolism is constructive metabolism, where we are going to take those raw materials, those smaller components, and we're going to build more complex molecules together. I like to think about this stuff kind of like Legos. There's a lot of similarities there, where catabolism is the breakdown of a larger structure. So if you acquired, say, a completed kit of some kind of Lego, you could break them up into smaller pieces. Anabolism is the act of constructing something new from those smaller pieces. So now, if you were to make something that you wanted to make that was not what that original structure was, you could build it using building blocks that you had just acquired from catabolism. Each cell has to perform thousands and thousands of different reactions all the time in order to stay alive and reproduce. These various chemical reactions that they use to accomplish these tasks are what we call metabolic pathways. Now, a metabolic pathway is a multi-step process where the organism starts with raw materials and changes them step by step by step with lots and lots of intermediate steps until eventually they end up with a completed final product. To understand what happens during this process, we have to talk about some important terms and get to know them. A substrate is a chemical reactant that enters into a metabolic reaction. So the various building blocks, the raw materials that are going to undergo metabolism, those are substrates. Intermediates are these middle step processes in the reaction. So if you took a snapshot at any point between starting a reaction and ending that metabolic pathway, any one of those snapshots would be an intermediate. A product is the end result of a metabolic pathway. It is the goal, it's the intended product, and also any byproducts that come from the process of this reaction, including things like waste materials. Those are still considered products of a reaction, even though it is not necessarily a useful one. Enzymes are the active moderators of these metabolic pathways. They are catalysts of a reaction. Catalyst is a really important term, a thing that facilitates a reaction without being used up in the process of that reaction. Again, really important term because an enzyme is a catalyst. Each step in these metabolic pathways is ultimately facilitated by a specific enzyme. There are many steps involved in many different metabolic pathways, and so for each one of those steps, you have at least one enzyme that moderates that process. Enzymes are going to vary a lot in terms of what their job is and also what kind of substrates they interact with. It's valuable to think about enzymes as lock and key mechanisms because enzymes are fairly specific in what they can interact with. Only certain substrates are going to actually be able to interact with a specific enzyme. When we talk about an enzyme and we talk about catalysts, they are facilitators of a reaction. It's important to note that these reactions, technically, they can happen without an enzyme being present. We don't have to have an enzyme in order to get a particular product. It's just that if you rely on these things happen naturally without the intervention of an enzyme, it usually requires considerably more energy, a strong amount of energy input than is usually available or common for this organism to have just kind of naturally in its body. That doesn't mean it can't happen. It absolutely can. It is rare and otherwise unpredictable. By providing an enzyme to bring these substrates together and hold them in just the right way to encourage this reaction, we can lower the amount of energy that is needed to facilitate the reaction. That means that we can make these reactions happen more often and on a schedule that is useful for a living thing. I mentioned a minute ago that enzymes bind to specific substrates, very similar to how a lock fits a specific key. Now, every enzyme has what we call an active site. This is the place where a substrate is going to come in and attach to. And this active site is very substrate specific. It has a specific shape that is going to match a specific substrate shape. It is going to be essentially the mold that this thing is going to fit into. Again, very similar to how a lock fits a specific key. And if you put a key that doesn't fit that lock in there, it might literally fit into the lock mechanism, but it won't turn. A reaction doesn't happen. Only when you have a substrate that actually fits that active site do we actually have a reaction take place. And so only for those substrates that can actually bind do they form what we call a complex with that enzyme. You end up with something that we call an enzyme substrate complex when the two things come together and attach. Only then can a reaction actually happen. And now here's an important thing to keep in mind with this lock and key model. The part of the key that matters here is the part that fits the lock. The substrate does not have to be exactly one key. In fact, you'll very rarely see an enzyme bind to only one known possible substrate. There can be multiple keys that can turn that lock. So for example, this key right here with the oval top has the same bottom part of the key, the part that fits the lock, as the one directly above it that has like the club symbol on it. But over here, we have two keys that don't match. We have another club key, but it doesn't have the same binding site on that substrate. It doesn't match the active site on the enzyme. Now, some enzymes can do all of this metabolic work they have to do without any additional modification. We don't have to change anything on that enzyme in order to make it function. Some, though, do have to receive a little bit of modification where they are naturally non-functional or less functional and have to receive something we call a cofactor in order to achieve a functional shape. Now, a cofactor is something that can fit into or attach to the active site of an enzyme to essentially make it achieve that final shape. And this can be a variety of things. There are, for example, we could have like inorganic ions like iron and magnesium and things like that. Or you could have organic vitamins and coenzymes that can come and fit in here. So a coenzyme is a small piece of a protein that can come in and fit with this larger enzyme. And an organic vitamin, while not protein based, will be otherwise an organic molecule. An apoenzyme is an enzyme that is naturally inactive, non-functional, until a cofactor arrives and attaches to it. So that cofactor is something that is required for function. Now remember, not all enzymes are apoenzymes, but for this group of enzymes, it is necessary for them to bind to a cofactor in order for them to achieve their final function. In this case, where they don't have a cofactor, the shape of that active site is distorted and warped, so it doesn't fit the intended substrate. In order for the substrate to be able to bind to that active site, you have to receive that special cofactor or coenzyme. Only then does the shape of the enzyme change to allow for that substrate to bind. This is an important reminder that an enzyme's shape dictates its function. And if you change that shape, you also alter that function. Now, cofactors typically have to be acquired from an organism's diet. However, there are some cofactors that can actually be synthesized. All coenzymes are synthesizable. They don't naturally occur in nature. We can actually make those, and we have to because they are protein-based. But cofactors that are, for example, those ions, those metals like magnesium and iron, and also certain organic molecules that are vitamins, are not always something that the organism who requires them can synthesize. So for some cofactors, that has to be acquired from diet. But for others, like those coenzymes and some of those vitamins, there are metabolic processes that exist for those organisms to help them build those things, mainly by converting one vitamin into another usually. Now, cofactor synthesis is really variable across species. Some species can make some of these vitamins and other ones cannot, and they have to be acquired from their diet. Now, some of these enzymes can also be enhanced or inhibited by additional binding sites that are present on that molecule. Allosteric enzymes are some such enzymes. They have these special allosteric sites that are present not where the active site is, but somewhere else on the molecule that special molecules called allosteric modulators can come in and attach to. And when these allosteric modulators bind to that enzyme, they can change the shape in one of two ways. They can act as inhibitors, where they alter the shape to prevent the binding of the substrate to the active site. So they actually distort the active site through attaching to and generally shifting the broader shape of this molecule, of this enzyme, to be something that the substrate no longer preferentially binds to. But they could also function as promoters, where they alter the shape to enhance and promote binding for that substrate. It might increase the effectiveness of that enzyme as a facilitator of that reaction. Now, these modulators are not required for enzyme function, however. They are not necessarily something that an enzyme has to have in order to function. That would be more along the lines of an apoenzyme. Now, let's talk about the influence of some of these environmental factors on enzyme activity, because this is going to be really important. We briefly talked about this in previous units. So let's talk about temperature first. Enzymes have an optimal temperature for their most effective and efficient function. If the molecule is at that specific temperature, they are functioning most optimally and their reaction speed is as good as it's ever going to be. But if you start to deviate from that optimal temperature, that is going to influence the reaction speed and the effectiveness of that enzyme. Now, if you move the temperature to be hotter, eventually, as you continue to increase that temperature, the protein is going to start to experience denaturation, where the enzyme is going to start unraveling. The bonds that hold the enzyme together in its specific shape, that starts to fail. And so the enzyme starts to unravel, lose its shape, and therefore lose its function. If you make the molecule colder, however, you don't get that same denaturation. Instead, the molecules just move considerably slower, and there is just less energy available to facilitate this reaction. What we experience is the rate of reaction changes and reduces, even though the enzymes still have their original shape. And eventually, if you get so cold, the reactions effectively stop. Now, shape is the decider for enzyme function. The lock has to fit the key. And enzymes get their shape by a variety of factors. This includes the sequence of those amino acids that are used to build that protein, of course, but also the interaction of those amino acids with its environment, especially with water and anything that it can use to form hydrogen bonds. Then you also have the interactions of the amino acids with each other, and some can form strong bonds, both hydrogen bonds and much stronger disulfide bonds. Now, high temperatures can actually break some of these bonds, particularly those hydrogen bonds. It can really mess with the way that these molecules interact with each other and with water. And as a result, they can change the shape of that enzyme and fully unravel it, and therefore the enzyme will start to lose its major function. Now, enzymes have a very similar relationship with pH as well, so acidity versus alkalinity. Deviations from that optimal pH are going to have similarly reduced efficiency, and we're going to see denaturation. But unlike temperature, where the proteins, mainly denature at high temperatures and they don't denature at low temperatures, if we deviate in any direction, high or low in pH, we're going to see denaturation. And this is because the pH impacts enzymes by messing with its ionizable amino acids. So essentially, we're messing with their charges. We're turning them into ions that change the way they interact with each other and with the environment. Ultimately, this also changes the enzyme's shape, just in a different way, and that similarly results in a change in function. Now, most enzymes function best at around a neutral pH across life, but this is true mainly for neutrophiles. Most life is neutrophilic, where we really prefer to live in mostly neutral environments, but that doesn't mean that an extreme specialist doesn't exist. We have extremophiles that love to live in acidic or alkaline conditions, acidophiles and alkalophiles. These species have evolved to be really tolerant to these extreme pHs, and so it is worth noting that in those conditions, those enzymes that they have a skewed optimal pH. So for a species that's an acidophile, its enzymes are not going to function best at a neutral pH. It's going to function much better at that typical acidic pH that it prefers to live in. It's worth noting that you see the exact same thing for temperature as well. That species that live in extreme temperature conditions, either very cold or very hot, their enzyme functionality is going to be shifted in that direction, where they're going to function best in the conditions that they mainly inhabit. Now, enzyme reaction rate is going to vary a lot with temperature and pH. As you increase or decrease those things, you're going to see a change in that reaction rate. But it's also worth noting that enzyme reaction rate will also change dependent on the concentration of that substrate. At low concentration, the reaction rate is pretty slow. And this is because there just isn't a lot of the substrate to get around. It's pretty unlikely for the substrate to encounter the enzyme. You got to remember that these substrates and these enzymes, they're not sentient. They do not think or want or need anything. They're molecules. And so what brings them together is usually the random movement of molecules as they are warm and they bounce around and they knock into other things. And when they hit other things, they ricochet off to hit other things. They bounce erratically and randomly through their environment. And eventually, just by sheer chance, a substrate and a matching enzyme are going to come together in just the right way to react. Inside of a cell, we increase the odds of this happening by making lots of enzyme and also providing lots of substrate and also confining them to a narrow space to make those interactions much more likely. As you increase the availability of that substrate, you make it more and more and more abundant, we're going to see that the reaction rate, the frequency of those interactions between the substrate and the enzyme, it's going to go up too, to a point. Eventually, the reaction rate's going to taper off because all of those enzymes that are available in that cell are going to be interacting with those substrates.